Hi all, I'm THERISSA from Andhra Pradesh, India. And first of all, I am very grateful for my home university, Acharya N.G. Ranga Agricultural University, Guntur, for selecting me under International Institutional Development Plan and sending me to Malaysia. And this is the petty details of my industrial training program in Malaysia for 1 month. And this is about the arrival. I have arrived to Kota Bharu on 14th February 2023. We had an opening ceremony and the faculty has delightfully welcomed us. These are the pics of our arrival. And coming to introduction, these are all my details. And coming to location of training site, this is the site where we have worked for this 1 month. This is the University of Malaysia campus, which is located in Jali, and working at training site. So basically we have worked on the concept of plant tissue culture. So what is a plant tissue culture? Plant tissue culture is defined as cultural plant seeds, organs, explants, tissues, cells or protoplasts on a chemically defined synthetic nutrient media under sterile and controlled conditions of light, temperature and humidity, etc. So you might be awestruck by the amazing power of the tissue culture. I can say that in one sentence that it is the generation of the enormous plant from a single tiny cell. And we also have a plant tissue culture lab. What are the components of a plant tissue culture lab? Every plant tissue culture lab must have these basic environments like aseptic transfer room, washing room, media preparation room, culture room, and an office. And basically we have worked with orchids. So what are orchids? Orchids are plants that belong to the family Orchidaceae, a diverse and widespread group of flowering plants with blooms that are often colorful and fragrant. And these are the different types of orchids that I have worked with, like Oncidium, Dendrobium, Spartaglottis, Vanda, Phalaenopsis, etc. And what is so special about an orchid plant? Orchids are one of the largest genera of the flowering plants with about 30,000 species worldwide, and they are also vary widely in their appearance. Many have stunningly beautiful flowers with stripes and spots. And I have been allotted with the Dendrobium variety of orchids, and these are the details of the Dendrobium orchid. Dendrobium is a genus of mostly epiphytic and lithophytic orchids in the family Orchidaceae. It is a very large genus containing more than about 1800 species that are found in diverse habitats. So these are also sympodial herbs with cylindrical roots usually arising from the base of a pseudobulb. And these are the list of activities that I have performed in our internship program, like subculturing of explants of Dendrobium, Oncidium, etc., acclimatization of contaminated jars, hardening of explants, media preparation and autoclave, ginger sowing, tying up of plants in nursery, housekeeping of nursery, etc. So coming to the first, what is meant by subculturing of explants? Subculturing is nothing but the removal of the medium and the transfer of cells from a previous culture into a fresh growth medium, a procedure that enables the further propagation of the cell line or the cell strain. This is the process of subculturing, and these are the pics of my subculturing. And the next is acclimatization. Acclimatization is a process in which an individual organism adjusts to a change in its environment, allowing it to maintain fitness across a range of environmental conditions. Usually, when the plants which are to be grown in soil are grown in jars, there will be some difference in temperature, light, humidity, rate of respiration, photosynthesis, etc., which will show an adverse impact on the plant growth. So, to make the plant suitable for the wide range of environmental conditions, we acclimatize the plant. So, this is a process of acclimatization, and these are the pics of my acclimatization. And the next is hardening of plants. Hardening of plants is nothing but the gradual acclimatization. For this, we have to transfer the acclimatized plants to the nursery. In the pots, we have to add some coconut chunks and also charcoal, and the acclimatized plants must be planted in that pots based on their sizes. These are the pics of hardening of plants. And the next is media preparation and autoclave. This is the basic and the most important process in tissue culture. This is the basic medium where the explants ought to be grown. So I have prepared murashis and scoop media for the Dendrobium species, and this is the process of the media preparation. After preparing the media, we have to keep in autoclave for 2 hours such that all the contamination is killed. And these are the pics of my media preparation. And the next activity is ginger sowing. I have sown ginger in the nursery as a part of propagation. These are the pics of my sowing of ginger. And the next is tying off of plants. You know what? When the plant is growing due to the weight and also increased height of shoots and also roots, the plant tend to bend because of the weight, so and also due to gravity. So we tie up the plants on the beds in the nursery so that they stand erect in the nursery and grow erectly. So the final and last but not least is the housekeeping of nursery. I have participated in the activities performed in the nursery like cleaning trash, replanting of orchids, weeding, trimming, arranging the explants on beds, etc. So these are the pictures of my housekeeping of nursery. And finally, conclusion. And finally, I would like to conclude that I have seen the plants with my naked eye, which I have seen only in books. And I also like the punctuality of these people. So I'm very happy and proud and also grateful for getting this opportunity to have my internship program in a foreign. And also I can say only one thing, that to explore, explore, and explore, because you know only a drop of an ocean. Thank you.